this talk focuses on some of the more interesting experimental developments in mutation theory over the last few decades and how they might be better represented in models of population dynamics. So every model winds up being more simple than reality, but it's important to recognize when our original simplifying assumptions are no longer adequately capturing the observed dynamics, and when new modeling approaches need to be considered. In evolutionary theory, we've transitioned from the modern synthesis into the extended synthesis over the past few decades. And it's good to start by level setting some of the things we've learned about mutations as we've transitioned. First of all, the question is, do mutations have a functional bias? And the modern evolutionary synthesis arose in the 1940s, and they held that mutations were random with respect to fitness. Now, a lot of people used a different terminology for this, but essentially the idea is that mutations are outside of any control system and are essentially haphazard. But the question is, what would bias towards fitness actually look like? So a mutation is biased towards fitness if the bias of the mutation space overlaps functional space more than an arbitrary bias would. This requires knowledge of the available solution space, but the modern synthesis was actually solidified before the discovery of DNA, and therefore before the discovery of the genomic space within which mutations occur. Therefore, they weren't actually able to determine what the solution of space of evolution actually looked like. And so modern biochemical research on mutations as they occur in vivo point towards a bias towards fitness in many instances. So first I want to give you a quick intro to directionality. So let's consider an abstract search space that has three solutions. The success rate of a blind search in this solution space is just over 5%. However, we might have a bias search, but the question is it biased towards function? So here's a bias search, and in this search, the success rate is just over 12%. This is a directed solution space. Note that it doesn't require that there be a high solution rate. Here we are only hitting success 1 out of 8 tries. The point is that compared to the abstract space, this search is biased towards function. Note that there are also potential biases away from function. For example, if this bias was just a little bit different, it would drop the success rate to 0. Biases that are biased towards fiction are relatively rare compared to all potential biases. In any case, when I talk about bias towards function, this is what I mean in an abstract sense. So let's look at a few very obvious examples of mutations that are directed towards fitness outcomes. So we have lots of functional biases that have selection triggers. That's where the organism's environment is more or less a major control valve on the mutations that are generated. So the first example is arbutin in E. coli. So by default, E. coli cannot process arbutin, even though it has some cryptic genes that can be utilized for that. But the problem is that they lack a promoter. And however, if E. coli is starving and it's in the presence of arbutin, a mutation occurs that inserts a promoter that causes these cryptic genes to be activated. And this insertion sequence does not come about if both those conditions are not met, or at least the rate at which it occurs is orders of magnitude lower. So that's one example. Another example is in the somatic hypermutation of immunoglobulin genes. So in the immune system, the immunoglobulin genes undergo somatic hypermutation, and it's triggered by antigen presence, it's targeted by non-coding DNA, and it's mediated by enzymes which induce mutations. So in these cases, it's clear mutations are preferentially targeted at systems or outcomes directly related to stressors. But what about functional bias that doesn't have selection triggers? Well, first of all, we have to answer the question, what does it even mean to be biased towards fitness if the mutation is not triggered by selection? Well, a mutation is biased towards fitness if the bias of the mutation space overlaps functional space more than an arbitrary bias would. So if a mutation bias generally constrains mutations to occur in a manner consistent with the general semantic architecture of the genome, it's probably biased towards fitness. But this means that a systems biology view of genome architecture is important for understanding the mutation system. We have to understand how the genome works from a semantic perspective, from understanding it as a system as a whole in order to understand how the mutation system is operating. So an example of this is pilin antigenic variation. So pilin is an outer coat protein used in Neisseria. It has an expression locus, pil-E, as well as multiple silent pseudogene loci called pil-S. And these pseudogene loci contain different versions of the pilin gene. And so mutation in this system occurs by a one-way non-reciprocal recombination from the storage version, one of these pseudogenes, into the expression version. And so since it's a one-way recombination event, we maintain the existing storage version while altering the expression version. So because the recombination is one way, the repertoire of alternatives is maintained indefinitely. But these are all functional pilin genes. So this process is not stress-induced, it's constitutive. So it is occurring whether or not there is selection pressure to do so. But because all the variations of this are still pilin, the mutations are biased towards fitness in general, but not necessarily towards the specific environment. All right? So they are biased towards the general function of being pilin, even though they are agnostic towards what the mutation is agnostic towards what the outer environment looks like. So now I'm going to take a shift and talk a little bit about cyclical mutations. So mutations are often thought, at least implicitly, as being permanently changes to the genetic code. And so when modeling, there's some general background assumptions that are commonly used. The assumption that mutations are not controlled by cellular mechanisms, that mutations are rare and distributed, and that for a given mutation, a back mutation becomes statistically improbable noise. And even when they occur, the genetic context of a back mutation will likely to have changed sufficiently to make it not have meaningfully reversed its behavior, even if the sequence has reverted back to an original sequence. But if we revisit these assumptions, as we've seen, cellular machinery actually does contribute to mutational control. Those examples that I showed, there actually are instances where mutations are controlled by cellular machinery. So we can't assume that they're not. The idea that mutations are rare and distributed, other evidence has shown us that no, there's actually hot spots of activity where the mutation rate is actually orders of magnitude higher. But because of that, because of hotspots and specific genetic structures, which we'll talk about in a moment, they can make back mutations relatively common. So that makes mutations cyclical rather than directional. And then what we'll talk about in the next section is when mutations are occurring in a way consistent with the genomic architecture, the behavior of specific previous sequences can be preserved even as other mutations accumulate. So let's talk about these mechanisms of cyclical mutation. So the first one is the one we've talked about so far, where we have a one-way recombination. So in Neisseria, you've got this repertoire of alternate coding genes that are alternate codes for the gene, and it is cycling through these different alternative versions. So that is so you frequently revisiting prior states based on this cycling mechanism. You also have transposable elements. Transposable elements can be inserted, but they can also be excised, depending on the transposable element. And so for transposable elements that have enzymes for both insertion and excision, that operates as a cyclical mechanism, where you're revisiting previous states. One of the more interesting ones is simple sequence repeats. So a simple sequence repeat mutates almost entirely by copy number variation. So it mutates by increasing or decreasing the number of repeats. And so because of that, you frequently revisit copy numbers because it varies by expanding and contracting. And then phase variable genes oftentimes mutate by promoter DNA inversion. So you have a promoter sequence, and that promoter sequence can be reversed to turn the gene off in different generations. So this promoter gets, sometimes it's forward and turns the gene on, sometimes it's backwards and turns the gene off. So again, you continually revisit prior states. So the question is, are cyclical mutations just an interesting sideshow? Well, it turns out they actually might predominate, at least in short-term genetic variation. So for a single type of cyclical mutation, these simple sequence repeats, even though they're a fairly small fraction of the genome, they actually mutate at an extremely high rate. In fact, they can actually be the predominant mechanism of mutation in a cell. So for the human genome, we found that the load of de novo SSR mutations is at least 75 mutations per generation, rivaling the load of all other known variant types. All right, so now I want to talk about another aspect of cyclical mutations called parameterized mutations. So what is parameterization? So a parameterized function is one where the mechanism of the function is controlled based on one or more adjustable variables known as parameters. Now, usually for something to be a parameter, its value needs to maintain a logical relationship with what it's adjusting. So for example, setting a thermostat. The set value for the temperature is a parameter, and the mechanism's function varies in a meaningful way based on the parameter. So the question is, what if mutability of genomic space tracks biological or environmental parameters? So parameterization is where mutation meets genomic structure and systems biology. And Caporal calls this the implicit genome. And we actually have many examples of parameterization in biology, and we keep on finding new ones. We have parameters that are acting like a binary switch. We have parameters acting as discrete selectors, or in the case of simple sequence repeats, the length operates as a semi-continuous numeric value, kind of like a tuning knob or a dial. Each of these adjusts a parameter of biological function in a way that is regularized with the phenotypic effect. You can see in this table all sorts of examples of parameters that have been found in the molecular biochemistry literature. So a visualization of this is here. So you have on the left, you have parameterized mutations. You basically have well-defined slots for changes to occur in, which allows us to easily revisit those same slots. And those slots of function maintain their meaning even when other slots are changing. On the right, you have the notion of mutation from the modern synthesis, where you basically have a functional system and you're making kind of an arbitrary rather than a parameterized change to it. And that change might wind up being good, it might wind up being bad, but ultimately it's just kind of an arbitrary change. And I want to note that both kinds of mutations occur in biological systems. The point that I'm wanting to drive home is the distinction between these is important because each kind has vastly different dynamics. All right. And so as we investigate mutational mechanisms of mutations in greater and greater biochemical detail, we keep on finding more and more examples of parameterized systems. And these parameterized systems benefit the organism in many different ways. First of all, they reduce pleiotropic disruption by constraining variation within the genomic architecture. Right? So they're occurring in a way that's consistent with the other parts of the genome. They're producing closer correspondence between mutational variation and environmental variation. And they're creating stronger links between the mutational biochemistry and functional outcomes. However, parameterized evolution requires different approaches to modeling in order to capture population dynamics. So next I want to talk about modeling. So how are we going to model parameterized mutations? Well, we're basically going to create a set of parameters. We're going to create an implicit genome, and we're going to create a set of organisms that have values for those parameters. And then we're going to create a set of environments that have how those values map to fitness within that environment. And so if you're interested in running this yourself, I have the link to the code below, and you can check it out yourself. So this is what it looks like. So we have an implicit genome, and we have both continuous and discrete parameters defined in our genome. And then we have a set of organisms which have values for those organisms. And then you can see the set of environments, each that have a fitness value for different parameters, or where the maximum fitness occurs on the value set. And so then we grow and evolve our organisms in each of these environments, and occasionally we switch environments and we evaluate the dynamics of what's happening. And the dynamic we're particularly interested in here is the ratio of beneficial to deleterious mutations. So what we found is that as organisms persist in an environment, the beneficial deleterious mutation ratio declines. But then we have a sharp uptick in the ratio during environmental switches. So the surprise is that a low beneficial deleterious ratio does not mean that the organism is suffering from the genetic load. It just means that it's near an optimal spot in its environment. Because the mutations are cyclical, mutational load is easily reversible or counteracted by either back mutations or environmental changes. So kind of to explain what's happening, it's easiest if we simplify this to an overly simple model. So in this overly simple model, each organism will have exactly one gene, and this gene will have one of four possible states. Basically, you can think about the Neisseria example, we have discrete values for discrete codings for your gene. And so these four states are A, B, C, and D. So when we begin the simulation, each genotype has the same population size. But because the fundamental theorem of natural selection, whichever one has the highest fitness will wind up dominating the population. So in this case, we're using B as the one that has the most fitness. And so you can see that the population of B explodes compared to the other ones. But that means that most mutations are going from B to other configurations, which means that they are deleterious. So that most mutations are going to be numerically are going to be away from this high fitness genotype, and therefore they'll be deleterious. But when we have an environmental change, you'll note that B is still the most numerous at this present moment, but it's not the most fit. And so therefore, the mutations away from B, some of them are going to be going to higher fitness states rather than all of them going to lower fitness states. And because of this, the beneficial deleterious ratio will jump higher. Thus, for parameterized mutations, as organisms persist in an environment, the beneficial deleterious ratio of mutations should drop, with the ratio rebounding on an environmental change. And I want to take a quick note on what this means for the concept of deleterious mutations. So in parameterized mutations, mutations can still be deleterious, but this deleterious nature represent a temporary environmental mismatch, not a degradation of the genomic architecture. So the recovery from such deleterious parameterized mutations is regularly and easily done by back mutation and environmental change, and one that's frequently overlooked, simple migration. If an organism doesn't match its environment, it can move. So consequently, deleterious parameterized mutations do not contribute to genetic load in the same way as non-parameterized mutations. So while the concept of deleterious is a valid experimental category for fitness effects, its meaning for evolution is ambiguous without further understanding the relationship of the mutation to the genomic architecture. And this is why a systems biology view of the genome is so important for understanding the process of mutation. Thank you very much. If you're interested in this, you can scan the QR code to get these slides and some additional related content. Additionally, if you want to ask questions, there's a link in there to ask questions, and I'll try to get back to you. Thank you very much.